Section 11 of Gray's Anatomy Part 4. This is a LibriVox recording. All LibriVox recordings are in the public domain. For more information or to volunteer, please visit LibriVox.org. Recording by M.L. Cohen. Anatomy of the Human Body Part 4 by Henry Gray. The Hind-brain Part 4. Microscopic appearance of the cortex. The cortex consists of two layers, that is, an external gray molecular layer and an internal rust-colored nuclear layer. Between these is an incomplete stratum of cells which are characteristic of the cerebellum, that is, the cells of Purkinje. The external gray or molecular layer consists of fibers and cells. The nerve fibers are delicate fibrillae and are derived from the following sources. A. The dendrites and axon collaterals of Purkinje cells. B. Fibers from the cells of the nuclear layer. C. Fibers from the central white substance of the cerebellum. D. Fibers derived from cells in the molecular layer itself. In addition to these are other fibers, which have a vertical direction and are the processes of the large neuroglial cells situated in the nuclear layer. They pass outward to the periphery of the gray matter, where they expand into little conical enlargements, which form a sort of limiting membrane beneath the pia mater, analogous to the membrana limitans interna in the retina, formed by the sustentacular fibers of Mueller. The cells of the molecular layer are small and are arranged in two striata, an outer and an inner. They all possess branched axons. Those of the inner layer are termed basket cells. They run for some distance parallel with the surface of the folium, giving off collaterals which pass in a vertical direction towards the bodies of the Purkinje cells, around which they become enlarged and form basket-like networks. The cells of Purkinje form a single stratum of large flask-like shells at the junction of the molecular and nuclear layers, their bases resting against the latter. In fishes and reptiles, they are arranged in several layers. The cells are flattened in a direction transverse to the long axis of the folium, and thus appear broad in sections carried across the folium, and fusiform in sections parallel to the long axis of the folium. From the neck of the flask, one or more dendrites arise and pass into the molecular layer, where they subdivide and form an extremely rich arborescence, the various subdivisions of the dendrites being covered by lateral spine-like processes. The arborescence is not circular, but, like the cell, is flattened at right angles to the long axis of the folium. In other words, it does not resemble a round bush, but has been aptly compared by Obersteiner to the branches of a fruit tree trained against a trellis or a wall. Hence, in sections carried across the folium, the arborescence is broad and expanded, where in those which are parallel to the long axis of the folium, the arborescence, like the cell itself, is seen in profile and is limited to a narrow area. From the bottom of the flask-shaped cell, the axon arises. This passes through the nuclear layer and, becoming medullated, is continued as a nerve fiber in the subjacent white substance. As this axon traverses the granular layer, it gives off fine collaterals, some of which run back into the molecular layer. The internal, rust-colored or nuclear layer is characterized by containing numerous small nerve cells of a reddish-brown color, together with many nerve fibrils. Most of the cells are nearly spherical and provided with short dendrites which spread out in a spider-like manner in the nuclear layer. Their axons pass outward into the molecular layer and, bifurcating at right angles, run for some distance parallel with the surface. In the outer part of the nuclear layer are some larger cells of the type 2 of Golgi. Their axons undergo frequent division as soon as they leave the nerve cells and pass into the nuclear layer, while their dendrites ramify chiefly in the molecular layer. Finally, in the gray substance of the cerebellar cortex, there are fibers which come from the white center and penetrate the cortex. The cell origin of these fibers is unknown, though it is believed that it is probably in the gray substance of the medulla spinalis. Some of these fibers end in the nuclear layer by dividing into numerous branches, on which are to be seen peculiar moss-like appendages. Hence, they have been termed by Raymond y Cajal the moss fibers. They form an arborescence around the cells of the nuclear layer and are said to come from fibers in the inferior peduncle. Other fibers, the clinging or tendril fibers, derived from the medullary center, can be traced into the molecular layer, where their branches cling around the dendrites of Purkinje cells. They are said to come from fibers of the middle peduncle. 2. The independent centers of gray substance in the cerebellum are 4 in number on either side. One is of large size and is known as the nucleus dentatus. The other 3, much smaller, are situated near the middle of the cerebellum and are known as the nucleus emboliformis, nucleus globosus, and nucleus fastigiae. The nucleus dentatus is situated a little to the medial side of the center of the stem of the white substance of the hemisphere. It consists of an irregularly folded lamina of grayish-yellow color, containing white fibers, and presenting on its anterior medial aspect an opening, the hilus, from which most of the fibers of the superior peduncle emerge. The nucleus emboliformis lies immediately to the medial side of the nucleus dentatus and partly covering its hilus. The nucleus globosus is an elongated mass directed anterior-posteriorly and placed medial to the preceding. The nucleus fastigiae is somewhat larger than the other 2 and is situated close to the middle line at the anterior end of the superior vermis and immediately over the roof of the 4th ventricle, from which it is separated by a thin layer of white substance. The cerebellum is concerned with the coordination of movements necessary in equilibration, locomotion, and prehension. In it terminate pathways conducting impulses of muscle sense, tendon sense, joint sense, and equilibratory disturbances. With the exception of the ventral spinocerebellar fasciculus, these impulses enter through the inferior peduncle. The reflex arc is completed by fibers in the superior peduncle, which pass to the red nucleus and the thalamus, and thence, by additional neurons, rubrospinal tract, to the motor centers. The exact function of its different parts are still quite uncertain, owing to the contradictory nature of the evidence furnished by 1. Ablation experiments upon animals, and 2. Clinical observation in man of the effects produced by abscesses or tumors affecting different portions of the organ. The 4th ventricle, ventriculus cortus. The 4th ventricle, or cavity of the hindbrain, is situated in front of the cerebellum and behind the pons and upper half of the medulla oblongata. Developmentally considered, the 4th ventricle consists of 3 parts: a superior belonging to the isthmus rhombencephalon, an intermediate to the metencephalon, and an inferior to the myelencephalon. It is lined by ciliated epithelium and is continuous below with the central canal of the medulla oblongata. Above, it communicates by means of a passage termed the cerebral aqueduct with the cavity of the 3rd ventricle. It presents 4 angles and possesses a roof or dorsal wall, a floor or ventral wall, and lateral boundaries. Angles. The superior angle is on a level with the upper border of the pons and is continuous with the lower end of the cerebral aqueduct. The inferior angle is on a level with the lower end of the olive and opens into the central canal of the medulla oblongata. Each lateral angle corresponds with the point of meeting of the brachia and inferior peduncle. A little below the lateral angles, on a level with the striae medullares, the ventricular cavity is prolonged outward in the form of 2 narrow lateral recesses, one on either side. These are situated between the inferior peduncles and the flocculi, and reach as far as the attachments of the glossopharyngeal and vagus nerves. Lateral boundaries. The lower part of each lateral boundary is constituted by the clava, the fasciculus cuneatus, and the inferior peduncle. The upper part by the middle and the superior peduncle. Roof or dorsal wall. The upper portion of the roof is formed by the superior peduncle and the anterior medullary velum. The lower portion by the posterior medullary velum, the epithelial lining of the ventricle covered by the telechoroidea inferior, the tanae of the 4th ventricle, and the obex. The superior peduncle, on emerging from the central white substance of the cerebellum, pass upward and forward, forming at first the lateral boundaries of the upper part of the cavity. On approaching the inferior colliculi, they converge and their medial portions overlap the cavity and form part of its roof. The anterior medullary velum fills the angular interval between the superior peduncle and is continuous behind with the central white substance of the cerebellum. It is covered on its dorsal surface by the lingula of the superior vermis. The posterior medullary velum is continued downward and forward from the central white substance of the cerebellum in front of the nodule and tonsils, and ends inferiorly in a thin, concave, somewhat ragged margin. Below this margin, the roof is devoid of nervous matter, except in the immediate vicinity of the lower lateral boundaries of the ventricle, where 2 narrow white bands, the tanae of the 4th ventricle, ligulae, appear. These bands meet over the inferior angle of the ventricle